So welcome all to the UNI.MINDS Festival. Once again, for those who came out from the panel, from the previous panel, thank you for choosing this parallel session, Gene and Cell Therapy. This event is organized by the Knowledge and Technology Transfer Office of the Universities of Ljubljana and Maribor, in collaboration with industry partners and supporters from the innovation community. The main partner for this year's event is Novartis Slovenia, but there are other supporters too listed here. This event will not be possible without the support of the European Union and the Republic of Slovenia under the European Regional Development Fund. I am Gabriela Droga and will be sharing this session with you. In this parallel session, we will be giving you a snapshot of innovative projects and good collaborative practices in the field of gene and cell therapy. Each speaker has a 3-minute long presentation, followed by 3 minutes of questions and comments. I'm going to notify you when you have reached the time limit. And in the end, our moderator will encourage you to have an open dialogue with us towards the future development trends in the field. Let's briefly go over some technical stuff. So first I ask you to change your view to a gallery view. So you choose side-by-side gallery mode. Also, please check out that your name is your name for us to know who you are. And throughout the event, we encourage you to have your camera on so that we can see each other. And to post your questions on the chat box from Zoom. Later on, in the end of the presentations, you can also ask the questions yourself. Also, a reminder to all speakers and attendees, you can use the UNI.MINDS themed backgrounds on the link in the chat box. So now I am very happy to present our moderator, Dr. Damijan Miklavčič. I hope he's with us already.
Yes, I am.
Okay, so thank you for being with us. He is the CEO of EduCell, the biotechnology company that has for more than 20 years been providing devoted service to patients on the field of cell therapy. He has extensive experience in SME founding and managing, and he's advisor for Technology Park Ljubljana and member of scientific advisory boards of several biotechnology companies and research projects. So I'm very happy that you are with us, and that we can facilitate this conversation, this dialogue with our guests.
Thank you, Gabriela.
So maybe if you, I will introduce. Do you introduce the first speaker, or shall I do it?
First speaker should be by
Damijan Miklavčič.
Damijan Miklavčič. I can introduce it just shortly, and then not to spend too much of his time. He's experienced life science professor and scientist with the field. We are collaborating also together for several years, and he has a very broad experience in electroporation technology, which is linked with the biology, medicine, and so on. And he has a lot of translational experience in the field. All the rest he will tell by himself. So thank you, Damijan.
Well, thank you very much. I think I need to share the screen. Hopefully this is, you see the screen?
Not yet. You have to share.
Yeah, exactly. So again, I need to select, need to select this. I went too quickly. So hopefully this is now visible. Yes.
Yes, you can put it on. It's okay.
Yeah.
Just go into a slideshow mode.
Okay. So here we are. So As Mia said, electroporation is a key word that I'm going to be talking about, and so I think the first thing first. Although some of you may know what electroporation is, or you may have even used it in your studies, research, or during the formation. When a cell is exposed to the electric field, what we can observe is a transient increase of plasma membrane. And this is allowing molecules to be introduced into the cells, but also to be extracted from the cells. Under controlled parameters, we can achieve reversible electroporation, meaning that the cell survives. Of course, we can also kill the cell, which is also useful. For 25, almost 30 years now, we have been working together with Gregor Sersha and other colleagues from the Institute of Oncology and from around the world to introduce small molecules for electrochemotherapy that is enhancing the locally chemotherapeutic effectiveness in cancer treatment, and specifically in solid tumors. So we have gained, I would say, a great deal of knowledge on different levels of biological complexity, but also engineering knowledge has been developed, including devices and models. As Mio mentioned a few years ago, we started to use electroporation together with them in order to facilitate cryopreservation without actually using current standard DMSO as the cryoprotectant, and we were quite good actually. So we showed this also with different sugars. Large molecules, on the other hand, can also be introduced for gene therapy and DNA vaccination. In fact, 2 years ago, there was a paper in Nature where they showed that CRISPR-Cas9 genome was used to modify T cells. Although this was done ex vivo, and they don't really mention electroporation in the abstract, the paper actually contains word electroporation. So we can introduce RNA, DNA as vaccine, but also for gene therapy. There's over 6,000 diseases with identified underlying genes that can be targeted, that can be replaced at this stage. So learning from oncology and introducing small molecules into cells and cells in tissue, we have also learned that in vivo, actually, in tissue, we see much more than simply cell membrane electroporation. We see, first of all, an inhomogeneous electric field distribution, which depends on isotropy. We see around the electrodes pH changes, even thermal damage can occur. But we also see blood flow modification, we see increased tissue porosity, and we see immune response. Now, we've done a lot of work, and we are, of course, continuing research, but we also collaborate with Slovenian companies, but also with multinational companies, most notably with Medtronic in the last 5 years. And I think this is something that I have over time already. So thank you very much.
Thank you, Damijan.
Thank you. Yes.
Leon, if you had some question, comments.
Yes, the thing is, we have just one short comment or question. I would like to give your short opinion. What is the electroporation technology compared with other methods to enter, let's say, DNA or some other drugs into the cell. So what is your opinion, how it is, let's say, efficiency compared to efficiency? What is the future of it in the, let's say, near future?
Well, thank you, Mia. I think this is actually, first of all, I'm biased. I'm working for 25, 30 years in electroporation. So what I'm going to say is, of course, electroporation is the way to introduce DNA into the cells. And we know that for in vitro. And it's, I mean, nucleofector is electroporation, just so that everybody understands that nucleofection is not something else. It's electroporation. So we can be very successful in vitro. What we are lacking right now, and this is something that needs to be improved, is the efficiency in vivo. And there are, of course, necessary steps that we need to do in order to bring the efficiency of electroporation gene transfer close to the efficiency of the viral vectors. And the benefit of that is that electroporation is not size limited in terms of how large plasmid you can introduce. So CRISPR-Cas9 actually is too large to be introduced by adenoviral vectors. So there's a lot of people who are strongly believing that CRISPR-Cas9, the future of CRISPR-Cas9 is linked to electroporation. And the other thing is that, of course, it's much less, I would say, hidden danger in immune response or immunotoxicity if you use electroporation compared to viral vectors.
Okay. Thank you very much. I think we have to stay on time, so there will be some time later on to discuss. And the next speaker is Professor Robert Zorec. Is the agenda is okay, Gabriela?
Yes, it's fine.
So it's again, you know, a person which we collaborated quite a lot, so we know each other. But he's a professor, he's entrepreneur, he's also a member of Slovenian Academy of Science. And I would like that he presents one very important translational approach of science in university and in his company. Robert, please.
Yes, can you hear me? Thank you very much, Mio.
Yes, no problem.
A warm welcome to everybody who's listening to this event. So I would like to talk about precision medicines, and we started to actually develop these medicines in 2009 when European legislation started the new regulation on advanced therapy medical products. And among these are also cell-based products, and I'd like to just bring forward some news about advanced personalized cell-based medicine to treat prostate cancer. And here are my colleagues that are part of this project, and I'm just merely humbly presenting this data. So the idea of this project was that, just give you some background. So our basic science project that we developed is related to understanding membrane fusion, the physiology of it. For this, we developed a number of projects, methodology from electrophysiology to super resolution microscopy. And after we published over 200 papers and got patents, we decided to go on a translational sort of event to develop this precision medicines. One project that I will not talk about is developing small molecules that target neuroglia. And the other project, as I already introduced in precision medicine, is advanced cell-based medicinal product. It's somatic cell immunotherapy for cancer. So this hybrid cure project, as we call it, actually also somehow coincided with the starting up of a company at a tech park, and where the GMP facilities are based, where actually we can produce these advanced cell-based medicinal products for clinical trials. Now, once we have decided to go for a advanced cell-based medical product, as Damijan Miklavčič presented, we used electrofusion to fuse two cell types: cancer cells, as you see here, and dendritic cells. Dendritic cells in the body are presenting antigens, and antigens also those that are hidden by cancer cells, but only if they are fused together. So this technology was developed by designing a biocompatible sort of equipment. And then the idea is to actually inject these immunohybridomas in the scenario of prostate cancer, to actually take a biopsy from the prostate, and then of course this tumor biopsy is used to generate a suspension of cells. And at the same time, we take blood and isolate leukocytes, monocytes from which the dendritic cells are produced, and then we fuse them. The tumor cells and dendritic cells are fused, giving immunohybridomas, and immunohybridomas are then injected back into dermally, hopefully to actually teach the immune system what are the antigens that the T cells have to attack that are tumor cells. So having developed this technology, we decided to go on an outpatient basis treatment of prostate cancer at the University Medical Center, Department of Urology. And here are just the results, how successful this therapy is. It's very encouraging. So if we measure time to next therapy, that is, if a patient develops castration-resistant prostate cancer, that usually there is no treatment that has been very effective. And we see here a control survival curve of patients that were not receiving this advanced cell therapy, and time to next therapy for the group that actually has received this vaccination. And you can see a significant prolongation from 6 months to about 28 months, meaning that the data is very encouraging. And at the moment, we actually are ready to actually treat the patients under hospital exemption regulation. And of course, this technology is ready for tech transfer. And I would like to also mention the Smart Grid project that I will probably talk about in the next possibility. It is in a proof of stage concept where we have lead molecules and new receptors that target. And I'm happy to address any questions if there are. Thank you very much for your attention.
Thank you, Robert. Excellent presentation. And I would like to just ask you and probably stress that's very one part is very important. I think as far as I know, you are the only one who, or your group, who registered an ATMP as an hospital exemption. And in the specialized GMP facility, which is, I think, the only one such range in the region. And I would like to ask you to tell me what was, let's say, the main obstacles to go with such interesting therapy into the clinical practice.
Well, thank you. It's a long question and requires many minutes. But I would like to just say that the mindset when in 2009 new regulation in European Union was introduced, this generated a lot of questions about how to carry out from academia to, let's say, to actually healing, to actually using these medicines. Of course, this regulation was extremely complex and nobody understood it, not even the national agencies. But with some help, we were able to obtain these permits, and this was very, very, very fortunate. And so we have now this experience of how to bring across, an important task in translation, and that is not only have an idea and technology, but also to have permits. Because marketing authorization it's needed when you actually do treat patients with new drugs, and ATMPs are basically biologicals that actually and hopefully there is this exemption, hospital exemption that actually allows treatment, and we're going to be able to do that in the next year for the patients in Slovenia. Thank you.
Thank you. So we will have more time at the end of our session, so it would be good to take this at that time. I would, we are quite good on time. We are just 2 minutes behind. So I would like to introduce the next speaker, which is Uroš Petrovič from Institute Jožef Stefan. Who will present his presentation about multiplex CRISPR. So Uroš, please, unmute. Thank you.
Okay, thank you very much. Thanks for having me. So I'll talk about multiplex CRISPR. Now, most traits of living organisms, if not all, are actually affected by several genes. Even the so-called monogenic human diseases are actually polygenic if we consider also their recently discovered suppressive mutations. And this is why methods to change several positions on genomes are so important, and multiplex CRISPR is the best method currently available to such means. Now, the work that I'll present was done in my research group at the Jožef Stefan Institute and University of Ljubljana, and actually by 2 highly talented students shown here, Gašper Žun and Katja Dobršek. Now, our research group has been studying polygenic traits in yeast, so as a model organism, but our findings can and actually are often directly applied also to human genetics. Now, what is multiplex CRISPR? So this is a version of a CRISPR-Cas method, which I pretty much believe you are all familiar with, whereby several cuts, so more than just one double-strand breaks, are made in the genome simultaneously. And this enables genome editing at several locations at the same time. Now, in this experiment I'll share with you today, we put genes for 6 different guide RNAs, as shown here, on a single plasmid. And the idea was that the expression of these gRNAs will lead the Cas and the nuclease to 6 different positions on 5 different chromosomes in the yeast genome. And from another plasmid, then we have a Cas2, Cas9 expressed that creates cuts. And for each modification, obviously, we also have matrix DNA that enables DNA repair via homologous recombination. For clarity, this is not shown in the figure. Now, we selected these genes such that they're easily phenotyped, meaning that we can easily determine the effect of the CRISPR and thus measure the efficiency, which I will actually show in the next slide. So going to results now, what we often see is that with single guide RNAs, not only these 6 ones that we showed here, but also in other cases that we have been using CRISPR-Cas in yeast, we get 100% efficiency in yeast, or very close by to 100% efficiency. In this particular case, you can see that 4 of them were actually at 100%, 2 were not, but the average efficiency was 99, actually more than 99%. Now, when we increase the number of gRNAs and we increase the number of matrix DNA molecules, we expect it to have a decrease then in the efficiency. And while this actually plummeted when we went beyond 4 guide RNAs, so up to 5, or even 6 gRNAs, this was really too low to be useful. But with 4 gRNAs, we actually observed efficiency at 72%, which we think is a very good idea. And so, very good conclusion and very good outcome of this project. And so to conclude, I think I can say, or I think I showed you that the method that we developed actually enables modification, at least in yeast genome, of multiple locations at the same time. But actually, we expect that similar methods with modification that have been done from model organisms to human cells will not so far in the future be also available for even gene therapy of polygenic disease in humans. So thank you.
So thank you, Uroš. Can you please suggest how this will, let's say, change the current approaches of gene therapies. Can you predict how this can, let's say, be used in the near or medium near future? What are your predictions?
Well, as I said, so the main point is actually to address more than just one location, more than just one site. So even for monogenic diseases, we know that there could be several changes in the genome that have to be introduced, not just editing of a single locus. So that for sure is an opportunity how to modify several loci at the same point. But actually, I would say that in short term, as you were asking, I think it's more than actually gene therapy. It's more like a research tool, because you can have combinations of different alleles, and the point is that if you make subsequent single CRISPRs, the side effects that you introduce inevitably with each CRISPR event are actually lower here with the multiplex CRISPR, because it's actually just one experiment, actually just one procedure, even though it's at several locations. So I would say in terms of clinical research, this is definitely important research, and actually not only us, it's several groups working with multiplex CRISPR. But I think maybe not short term, but in mid-term, it will also be interesting for gene therapy directly.
Yes, but the problems which we see are validation of all these changes in the genome, and this is then a challenge for the developers. So thank you, Uroš. The next speaker is Jelka Pohar from National Institute of Biology. So please, Jelka, are you with us?
Yes, I am. Do you hear me?
Yes, but we don't see you.
Okay. Start. Yeah, but what about now? It's okay?
Yes, now it's much better, yes.
Okay.
Okay, please just start.
Okay, you see my presentation now?
Yes.
Okay. Okay, so thank you. I'm Jelka, and today I'm going to talk about the prospects of regulatory T cells for immunotherapy of autoimmune diseases. I will present our research performed at the Institute UNI.MINDS in Paris. And autoimmune diseases affect people of all ages and often greatly aggravating their quality of life. Unfortunately, the incidence and also prevalence are increasing, especially in the developed countries. Regulatory T cells are crucial for maintenance of homeostasis and prevention of autoimmune responses. They inhibit immune cells by direct or indirect mechanisms, such as expression of anti-inflammatory cytokines or via co-inhibitory receptors, and they can even promote tissue repair. Clinical trials demonstrated that polyclonal Tregs can delay or prevent graft rejection and also control autoimmune responses following adaptive transfer. Of course, antigen-specific Tregs are more effective, but such cells are rare and difficult to isolate. To circumvent this, we can engineer polyclonal Tregs into antigen-specific ones by T-cell receptor gene transfer. We have shown that Tregs engineered to express TCR, recognizing peptide from myelin sheaths in the central nervous system, that is SMOC peptide, protected mice from experimental autoimmune encephalomyelitis, which is a classical model of multiple sclerosis. You can see here. To understand how engineered Tregs work, we first generated mock reactive Tregs defective for production of IL-10, CTLA, or AREC, using retroviral vectors, additionally coding for siRNA. Silencing of IL-10 and CTLA-4 abolish the protection afforded by antigen-specific Treg cells, and also were not able to suppress expression of inflammatory cytokines such as interferon gamma from autoreactive T cells. Tregs thus protect recipient mice from disease through immunological mechanisms rather than tissue repair, rather by tissue repair. So antigen-specific Treg cells were able to intercept EAE progression when they were administered at the onset of the disease, but not at the peak of the disease. Also, engineered cells persisted in the animals by the end of the experiment, and compared to endogenous Tregs, still displayed activated phenotype and higher expression of proteins involved in suppression. To further develop cell-based therapies for autoimmune diseases and cancer, we will, with my good colleague, Dr. Angel Smolet, and with tremendous support from National Institute of Biology, establish a new lab dedicated to immunology and cellular immunotherapy. At this point, I would like to thank you for your attention and for inviting me to UNI.MINDS, and of course, I'll be happy to answer any questions.
So thank you, Jelka. Excellent. I congratulate you to join the Slovenian scientific community. I think we will all benefit because this field is increasingly important. We will have more time to later discuss about autoimmune diseases, but the thing is, do you see or foresee let's say, the new therapies coming from this research in the, let's say, in the next 10 years or whatever. So it means, as I see, I'm looking forward for the things, how they can be used in the translation. So do you see such an approach in the clinical, let's say, clinical application?
Yes, of course. And I think 10 years, it's quite a, yeah, I think it's quite a good time. In 10 years, I think something like this will arise. I mean, it's working in animal models, so therapy with antigen-specific Treg cells. For polyclonal ones, they are already used in some clinical applications, but for antigen-specific, yes, this is still in the development. But yeah, I think quite soon, because we also in Paris, we collaborated with team from University of Zurich, and they're actually discovering, especially for multiple sclerosis, they're discovering new antigens against which we can then target our Treg.
Okay, Jelka, thank you very much. We will continue then later. And now the next speaker is Dominik Gaser. So from Novartis, Slovenia. So please, Dominik.
Hello, can you hear me?
Yes, we can. It's fine.
I have problems with
and we see you too. So.
Super, great, but I have problems with, okay. Now it looks okay. Good.
It's fine. We see the presentation too, so it's fine.
Perfect. So I try. Okay, now. Yeah. So thank you for the introduction. I'm coming from LIC, which is a Novartis company, and Novartis is actually one of the pioneers which brought the cell and gene therapy through clinical trials and regulatory approvals and to the patients. And I want to give you maybe some pharmaceutical view and introduce shortly two available cell and gene therapies which are already on the market. So when we are talking about the cell and gene therapies, we can divide them into two big buckets, let's say ex vivo and in vivo. In case of ex vivo, the cells are taken from the body, they are then manipulated outside, and then when they are changed, they are going back into the patient body. In case of in vivo, the genes which are used for the treatment are injected directly into the patient's body. In case of an in vivo therapy, there is a gene therapy to treat spinal muscular atrophy. It is registered under name Zolgensma, and it is also available in European market now from April this year. Spinal muscular atrophy is diagnosed in early childhood, and if left untreated, it is the most common genetic cause of infant deaths. I would just shortly like to say how this treatment is prepared in pharmaceutical environment. Actually, you need mammalian cells which are transfected with several expression vectors which are coding for capsid of the virus. Then viruses are assembled in the cells, and when you have sufficient amount of the cells, these are lysed so that the viruses are released. And then in the purification process, you have to separate non-functional or empty viruses from the fully functional viruses, which could be used for the treatment. In the case of Zolgensma, actually, we are very glad that within Novartis Group, LEC was selected to conduct the medicine release testing for European market, which was a quite nice success of recognition of our work. Next example is ex vivo therapy. It's a CAR T cell therapy to treat acute lymphoblastic leukemia. In this case, the T cells are collected from the patient's blood. They are modified using lentivirus, so outside of the body CAR T gene is introduced into the cells so that the receptor is expressed on the cells, and then they need to be very fastly tested, controlled, and giving back to the patient. And what would I like to emphasize here, and it is very important that this is completely new approach in the treatment, and patient actually is involved in the complete circle of the medicine preparation, and it becomes very time critical from the point when the cells are taken from the body to the point when the cells are transplanted back into the patient's body. So in pharmaceutical industry has to develop the very reliable and fast testing techniques. So when the cells are generated, they need to be tested that they are safe, that they could go back into the body. And also here we contributed with our digital and next generation expertise, and we developed fast method for adventitious agent testing and identification. And what I want to say that although this contribution of our lab is really very tiny part of the whole development story of this cell and gene therapy, we are really glad that our expertise was recognized within the whole group, and that we are also able to contribute in this very interesting new field of therapeutics. So this is it from my side. Thank you.
Thank you, Dominik. Excellent. I congratulate you too, because I think that's quite an important, if let's say it's a big pharma is doing such an advanced therapy and in our neighborhood. So I think collaboration here is also very important for us. Short question. What was the biggest unmet need for you? What is the biggest let's say challenge, no problem, but challenge to let's say to finish such a project as you got it now? What you are, let's say, yes, the biggest challenge for you?
Yeah, actually, the biggest, it's not the expertise, you know, because we have strong experts, and they are mostly coming from the Slovenian university, but within such big pharmaceutical company, it's actually, and for the lab in Slovenia, it's actually quite hard to really position yourself also within such a big company that you are recognized, and that such important task as a European market testing for Zolgensma is, let's say, conducted in our lab. So, yeah, recognition and convincing the whole management and the structure in the company, it's not easy task, I have to say.
I can imagine. Okay, Dominik, thank you very much. We will come later on for a discussion about this topic. So the next speaker, thank you. Okay. So the next speaker is Kristina Orešič Bender from the Adverum Biotechnologies. So please, Kristina.
Thank you, Mara.
We see, just put it on the.
Yep.
Yep.
Okay. Thank you very much for the invitation. Thank you, Mimir, for taking the helm of this great conference. My name is Kristina Orešič Bender. I'm a principal scientist at Adverum Biotechnologies, a clinical stage company. We're developing gene therapy products for patients suffering from retinal diseases. We are a publicly traded company, so I have to disclose my forward-looking statements. If you go on the website for clinicaltrials.gov, you can see that there is over 180 interventional clinical trials that are involving AAV, adeno-associated virus, as a vector to deliver different kind of genes to patients. And majority of development is taking place in the eye. There is some products developed that are taking place like for development in liver, muscle, or brain. Majority of development is taking place like in phase 1 and 2 clinical trials, so relatively early stages of clinical development, where we are testing predominantly safety, but then we're also looking at the first efficacy window for the patient, trying to figure out what would be the clinical endpoints for phase 3 registrational trials. And really this field has bloomed in the past couple of years with the approval of 2 products in the past 2 years. Luxturna, that was approved in 2018, and Zolgensma, that Dominik just mentioned, that has been approved for patients that are suffering from SMA1 mutations, and it's a gene of mutation therapy, delivering SMA1 healthy gene to the patient. It's been developed by Regenxbio Biovexes and acquired recently by Novartis, while Luxturna has been recently acquired by Roche. So obviously there's so much interest and so much development going on in this field. We reached a point where something that was thought to be impossible just a decade ago is just really becoming possible. So where we are fitting in this particularly high development field as a company, we're really developing gene therapy products, not only for rare diseases, but also for prevalent diseases of the back of the eye, such as wet AMD or age and macular degeneration or diabetic macular edema. So these diseases are driven by pathological VEGF that is being secreted in the back of the eye. And this VEGF, vascular endothelial growth factor, actually is causing vascular leak in the back of the eye from the blood vessels in the back of the eye. And this vascular leak can cause interference in the vision and eventually vision loss if patients are not being treated. There is a standard of care for these patients. It's anti-VEGF therapy, and it's this therapy is like usually biologics delivered through regular monthly intravitreal injections. So it's injection into the back of the eye. And as long as the patients are getting these monthly injections, they are doing fine. So on this particular part of the slide on the left-hand side, we can see in green that patients in clinical trials are receiving on average 12 injections a year, so once a month. And on the y-axis, we're looking at best corrected visual acuity, which is how we measure the vision. And we can see that they're doing relatively fine. However, when they're not in clinical trials, when they're in the real world, so going to their ophthalmologists or trying to get to their ophthalmologists, there is a significant lack of compliance. And there is like a decrease in the number of injections that they're receiving. So from 12 a year to 4 up to 5 a year. And with that, there is significant decrease in the vision and even loss of vision. So lack of compliance is really a major obstacle like in the field and major obstacle for the patients to actually maintain and really like save their vision. And this is where we're coming in as a company. We're actually developing an AAV2 gene therapy product, which is using a serotype called 7M8, where we are delivering a gene therapy, by gene therapy, a standard of care molecule for VEGF-driven retinopathies. This is anti-VEGF molecule called aflibercept. So through a single intravitreal injection in the back of the eye of this gene therapy product, we are delivering cDNA for aflibercept. And now, like this particular virus is going to infect the cells in the back of the eye. And so by this infection and transduction, eventually, like the cells are going to become small factories and secrete aflibercept. And aflibercept can now efficiently suppress VEGF that is causing the pathology in the back of the eye. And when I was invited by Gabriela to participate in this conference, I was thinking a lot about like how actually academic research and innovation can help moving our field forward. There's so many obstacles that we are facing. You know, like we're a relatively small company, a little bit over 100 employees, and doing everything by ourselves. There's really like a lot of obstacles you can imagine. Large scale manufacturing, cost of manufacturing is really tremendous. And anything that can actually improve these processes would be tremendously helpful. There are difficulties with purification of AAV particles, obviously. And as Dominik was saying, vector quality control and assay standardization for the release of our product is really, really complicated. There's over 30 different assays that this particular product has to go through in order to be efficiently released and go to the patient. And I just put few as an example. For example, empty to full ratio. How many particles have DNA? How many don't have DNA? How many have partial DNA, for example? Assessment of the potency, DNA composition, capsid heterogeneity. And then all these assays had to be qualified and validated. So you can kind of imagine complexity of this release panel, and any kind of innovation within the methods like would be extremely beneficial. And then last but not least are these immunological barriers to AAV gene therapy, because AAV gene therapy is using a virus that per se is really highly immunogenic to our organism. So we actually have to find the right dose that can overcome this immunogenicity. So any kind of immune suppression of these immune responses would be extremely beneficial for the field. So this is where I see kind of that academic research can help us, like now when we have overcome this impossible to possible, to go even faster and deliver faster the gene therapy products to the patients with unmet medical needs. And then with this, I would like to stop and then continue hopefully with discussion, and looking forward to your questions.
Thank you, Kristina. Excellent. You really put some very important topics, which when we are developing science, we are not thinking about, but then this is the real bottleneck to use it. I have logically a lot of questions, but there is just one, which did you also try to ex vivo transduce the cells and put the already transduced and validated cells in the eye or not?
Yes, we have not like used transduced cells. Like we have a plethora of like cell-based assays for the release of the product. So that's like a first thing that you have to show, that you have a vector that can transduce cells and that the cells can actually express the protein of interest. Transduction, so cell therapy per se in the back of the eye is highly complicated, like concept, because eyes really per se such a complicated organ, right? So this is something that we just recently talked in the company, how you could regenerate the retina, like kind of like once the retina is really degenerating in the patients, like can you like kind of like regenerate the cells in the back of the eye and with that restore the vision, right? This is really nascent, like in nascency, these kind of concepts, but you know, maybe in a couple of decades we would get there. I don't know if that really answers your question. It's like highly philosophical. Yeah.
No, there are. We just pick one of the problems. There are thousands of them. So,
exactly. And like with AAV gene therapy, like just delivery, right? Like to really deliver efficient amount of therapeutic protein to certain compartment of your body is like such an extremely like challenging task, right? So we just got there, right? We're kind of like scraping on the surface, and we're kind of like riding on this wave of success of 2 products that have just recently been approved. So I think in the next couple of years, there will be many more products that are going to get approval.
Definitely. Yeah, and animal models and so on. So we will talk later about it. So thank you, Kristina, again. And we will move forward to Boštjan Vihar from University of Maribor, and he will present us 3D bioprinting, also a very important part in the regenerative medicine. So, Boštjan.
Yes, thank you for having me here and for the introduction. So I will be moving away from molecular biology for a little bit, so I hope it's still interesting. So I'm a postdoc at the medical faculty in Maribor, and in our department, our work is focused mostly on bio-based materials for controlled drug delivery and wound healing, but also establishment of primary cell cultures and the study of cell material interactions. So we recently started also exploring materials as...
Can you just put the PowerPoint into slideshow mode?
Oh, yes, sorry, of course. This should be better now, right?
Perfect.
Yeah, super.
All right, thanks. Yeah, so recently we started exploring the materials also as substitutes for extracellular matrix and 3D cell culture, and consequently also bioprinting. And today I would like to talk about the VitaPrint project, which is basically a collaboration between our department at the faculty and the company IRNAS, which is a technology provider for development of customized solutions. So basically what VitaPrint originated as was an assembly of more or less off-the-shelf parts, but also customized together to fit some of the for some of the applications that we were using for the assembly of tissue engineering scaffolds. And it slowly evolved into a more complex and now a really complete and customized system that it has a very high versatility and can be used for various applications. And I would like to take you through the process of how we basically collaborate on that kind of project. So what is really beneficial, I think, for both institutions in this kind of development is that on one hand, the technology provider can immediately optimize the technology for whatever applications we require at the faculty, but also on the other hand, at the faculty, we give direct user experience and feedback on what works, what doesn't work, what would be more necessary for us. So this really goes in many cycles, I would say, of design, development, testing, and then repeated iterations of that. And I would like to give you a concrete example on what we've been working at. So we started working on designing scaffolds for vascular tissue engineering at the faculty, for which we wanted to have tubular, so basically scaffolds which incorporated hollow channels that are perfusible and can be used to implement cells in a channel-like manner, especially endothelial cells to study growth and endothelial layer development, and later on also perfusibility and molecular transport. And for that, we required a special nozzle that would allow actually fabrication of channel scaffolds in a single step. And this is what you basically see here. This is the first prototype that we had that had biomaterial going out the sides, and basically a cross-linking solution that was liquid in the center. So you would have what we call a core shell filament that would be extruded with a solid and stable shell and a liquid core that was, after evacuating, would leave behind a hollow channel. And here you also see the first results, which were also published in the in Jove where we tested attachment and viability of endothelial cells. And now in the next level, we also want to integrate this together with, let's say, microfluidics, where we can control the composition of the scaffolds during the printing, so on the fly, and implement gradients and sequences, let's say, of different materials, of cell concentrations, and so on. Here on the red, on this side, you see basically the concept art for the next generation of the nozzle, which we have also now just manufactured the first prototypes. So we will be looking forward into testing them. So this is really more or less it. So if there are many questions, I would be happy to answer.
Thank you, Boštjan. Yes, this is for those who are working in cell therapy and regenerative medicine. We know that that's really the hot topic for the last years and also for the near future. And my question would be, did you already try to implant such device into the animal model, or are you planning to? Or
no, so at our department, we mostly work on in vitro models. And yeah, we want to, establish as complex ones as we can, but we haven't been working on implanting really. But this would be interesting to do in a collaboration, for example, with someone who has more experience.
Of course, yes. Okay. So thank you very much. At the end, we will join all these topics, and now we are moving, as far as I know, to the last presentation. And that's Vita Dolžan, who will, from the medical faculty, and she will present the pharmacogenetic approach in the cell and gene therapy. Vita, please.
Yes, hello everybody. I think I have a slight problem with sharing. Okay, you can see my screen, I think. So hello everybody. I would like to talk about opportunities and perspectives of translating pharmacogenomics and personalized medicine from laboratory to patients. And it is generally accepted that some patients do not respond to the drug treatment like the others, and such inter-individual variability in drug response may lead to adverse events, even to hospital admissions, or may cause death. More than half of these adverse events would be preventable, and leading to saving of hundreds or thousands of euros per patient. And a considerable extent of inter-individual variability in the response to treatment may be also due to genetic factors. And it has been shown that over 95% of European population carries at least one actionable genotype in a candidate pharmacogene. Therefore, pharmacogenomics has an enormous potential for improving safety of drug treatment, patient adherence, and healthcare through determining genetic variants in drug metabolizing enzymes, transporters, and receptors. There is sufficient evidence that pharmacogenomic testing leads to improved patient outcomes, and genotype-based dosing recommendations have been developed for over 100 gene-drug pairs. I have a privilege to participate in the largest European consortium in a Horizon 2020 project, Ubiquitous Pharmacogenomics, that led to development of genotyping approaches and clinical decision support tools that enabled systematic implementation of preemptive pharmacogenetic strategies across 7 European countries. nearly 7,000 patients have been involved in a crossover randomized clinical trial across Europe. In Slovenia, 716 patients. And currently, a robust assessment of how this intervention impacts patient care, treatment cost effectiveness, and quality of life is in progress. In future, ICT support and social innovation tools are needed to support transition from personalized drug prescribing to personalized healthcare. And pharma industry could also contribute by adoption of pharmacogenomic biomarkers in drug development and companion diagnostics, enabling of healthcare providers to introduce and adopt new healthcare paradigms is also important, and patients need to be empowered to become active partners. We are very happy to collaborate with this respect and to have a partner such as Health Lord. And I would like to acknowledge Gorazd Hladnik for his contribution and collaboration in this field of enabling ICT support and social innovation tools to translate pharmacogenomics into real action. So thank you for your attention.
So Vita, thank you very much. Yes, that's also another part which will bring personalized medicine, which should bring personalized medicine to reality. And I would like to ask you, you are experienced scientist and you have a lot of such projects in the past. But did you see let's say the challenge to introduce it into the clinical practice? Why, let's say, more of this knowledge, which is enormous now, is not used in the, let's say, daily business for the patients? Patients would be interested, definitely.
Yes, the main obstacle is that these guidelines that have been developed mostly by clinical pharmacologists and scientists, and are actually evidence-based for, as I said, more than 100 gene-drug pairs, they need to be translated or incorporated in the clinical guidelines of treatment, so that the medical doctors could use it in everyday treatment, not only within clinical trials or clinical studies. Further on, it depends a lot on policy decision makers. So we hope that this European project, Ubiquitous Pharmacogenomics, will really bring enough evidence that pharmacogenomic testing is cost-effective and efficient across different healthcare systems. And then also the reimbursement strategies need to be put into action.
Okay, thank you, Vita. Yeah, that's, I think that is a straightforward answer, and but I'm sure that there is a lot of work behind to really put it in reality. And so I wish you luck. So thank you very much. Thank you, everybody. Now it's time for questions. There are some on chat. There are some also behind the chat or in your heads. And I will just ask Gabriela, do you have any, let's say, support or
Yes.
what do you expect?
We had one question for Kristina before by Damijan, and I would suggest that all the speakers would come alive now, put your camera on, and maybe discuss lively this. You put a pertinent question, Damijan, to Kristina. Maybe this is a good starting point for the discussion.
Yeah. Well, I think the eye is immune privileged organs. So I think adenoviruses are not that, I would say, drastically to problematic. But if you look at what is happening in all the other treatments, I mean, in not in the eye, but if you go to other tissues where the immune response is much stronger, then you can be surprised, of course, the moment you move actually from one patient to another or to a different geographical location. So I think that makes the gene therapy so extremely difficult in one way. Yeah. So, but that's just my naive perspective.
Yeah, it's like you're totally right. It's so much easier. Obstacles are so much lower in the eye, right? It's not completely immune privileged. There is still like numerous quite different like immune responses there. But we have achieved that threshold where we can actually efficiently transduce without some kind of really detrimental effects on the tissues, unlike in the systemic tissues, right, where we know then once we have T cell activation there, we're losing the cells, we're losing the gene expression. So any kind of development and understanding immune responses and exactly what's happening, suppressing the right type of immune responses when you're delivering our gene therapy for the right period of time, understanding these regimens, whether they're going to be personalized or not, whether the patients have been previously exposed to AAVs or not, what the levels of potentially neutralizing antibodies or total antibodies to AAV they have. Like all these things will play a role for development of successful therapy. And although like, I really like Vita, your talk, it's really great. Like really kind of personal medicine is something that we all should be thriving for, right? We all want medicine that is tailored for us, for something that we need. However, it's so hard, right? We're all so different. We all have different level of expression of genes, exposure to different pathogens, and developing something that is really kind of tailored for all of us is a science fiction in terms of the costs, right? For any small company. So it's really, but I like to think about this because I think that's where we should be going, like in future, understanding how we can actually efficiently develop individualized therapies for everyone, and then still have successful company, right? So those are really good questions.
Yeah, thank you, Kristina. I would like to ask Robert, who I think is the only one who experienced the whole process of putting one therapy to really establish a therapy, going through the clinical trials, through all necessary steps. And what were, let's say, the biggest problems or for you, or challenges, what is modern to say, and how long does it take? You mentioned before 2009, but let's say, where were the biggest bottlenecks?
Thank you, Mia. The main problem, I think, is in the minds. That especially that somebody actually talked at the very high level at the medical school that we are too small to develop new drugs, which is not true. And anyway, if we would want to have like a nation to generate new drugs, that would be very easy, because if you have the will, you have the end result. So I would like to congratulate Dominik for this breathtaking, actually, really changes in the global actually map of gene and cell therapy to become kind of a global sort of lab testing this products, because releasing drugs for treatment is one of the key issues. And for that reason, when you step into the clinical trial, they want to have the highest possible standard, and that is the GMP standard that actually also academic groups have to develop. And this was a long process in European Union, where there was an initiative called academic GMP. That means that, why would an academic group need to have a GMP? because this is typically an environment that big pharma can afford it. And then gradually it came through that, in fact, even though this is an academic unit or lab, they need to have the highest standard of GMP. That's in the EU. In the FDA has a bit more pragmatic approach. And we decided to actually go through all these obstacles because of having experience working in a committee for advanced therapies. And that experience just was very, very important to see that if it's no big deal, you just say, we can go and we can do it, and then we go and do it. So, and as I mentioned, translation means not only understanding ideas and technologies, but also the mechanism of how to get the permit, the marketing authorization. And of course, 3 issues are very important in medicines. That is safety, efficiency, and quality. Mm-hmm. So this needs to be, of course, met with any development. Thank you.
Yeah, so thank you, Robert. Maybe just a comment. I was, just recently came across a presentation, and actually it surprises me that most of the 7 gene therapy are actually developed in small biotech company and academic environment, and then actually pharmaceutical industry step in when you need to, bring them to the patients, go through all these regulatory approvals and establish GMP testing. And also the investments are very different. So in the research, it's a lot of investment in the small and in academia, and pharmaceutical industry is investing really a lot into the studies and regulatory environment to make them accessible.
Yeah, and I can speak to it's not easy to raise money. Of course, it's extremely difficult. And like with a small company, like you might get to phase 1. We're in phase 1 for one indication, phase 2 for the other indications. So we're doing pretty well and have had a couple of rounds of very successful funding. Getting closer to phase 3s, we're talking about a couple of hundreds of millions of dollars. It's going to be much more challenging to fundraise, and then bigger partnerships will probably start taking place. But I love what Robert said. You are a champion if you have a good product, if you believe it, right? If you know it's going to work for your patients, and if you know that there is an unmet medical need, and you know how to have kind of vision how to bring that product to the patient, I think there is sky's the limit. There shouldn't be obstacles. You just have to keep on doing it, working it, build your community of colleagues, friends. Everybody kind of can cheer up for it and help you kind of build a company eventually, right? So even in small Slovenia, these things are really possible, and I'm so glad that things like that are happening.
Yes, I would like to mention this COVID crisis is an opportunity for our technology, because it's really making all these processes faster. And there is a need which has to be fulfilled, and you have an opportunity to try a lot of new things, logically according to these things what Robert mentioned, safety, efficacy, and so on. But if you don't measure, if you have patients who are already suffering, and if you are using scientific methods, which who knows them better than university and institutes, and small biotech who are somehow startups, then this can be, let's say, a more flexible way to come faster to the let's say, efficient treatment. But logically, if you want to spread it, the role of big pharma is important. They have the networks, they have their subsidiaries, they are covering the whole world normally, and they can put it everywhere if necessary. And this is good for the humanity, I would say, for the whole world population. That's excellent. I would like also to mention that institutes, some institutions in Slovenia, let's say like National Institute of Biology, then also Josef Stefan Chemical Institute, and so on, they were quite good supporters also on of this field, and they were involved in a lot of these techniques, also in the gene therapies, those which Dominik mentioned. and they used their infrastructure and the knowledge and developed the scientists who then supported and make these technologies happen. So that's an important part. And what I would like to ask then Jelka. Jelka, how you let's say recently returned back from France, and when you compare how it is done in the French very regulated state, how they run these collaborations and how they allow groups and small companies to come with their products or with their technologies on the market?
Well, I mean, for small companies, I don't. I mean, they usually emerge from these big institutes. Like, I don't think so. There was one at our institute, but for sure from Pasteur or Marie Curie Institute, they are like the biggest ones in Paris, for example. And yeah, they have support from the group they originate, and I think the technologies, the support, I think it's quite important. And I think they receive a lot of it, and they are attracting scientists also from USA, especially French scientists, to come back and to in one way to make good platforms which can then support this kind of company development, and on the other hand, yes, they attract a lot of good technologies, and it's really not I don't know, you have in one institute you have everything. You don't need to go to another institute, I don't know, to use one machine or the other. They have all the support, they have big platforms, and I think it's much easier for the scientists when they have such environment to develop their ideas and then also to commercialize it.
Okay. Okay.
If I can inject here, we have a question from David Dobnik. He will try to ask it directly, so he's unmuted and has a camera. David, please.
Yes, well, actually, it's not a question. Already written in the chat box. So perhaps this refers to the Dominik's part. We actually kind of developed and transferred the ddPCR method that will be now used as a release testing in the Vaxis labs. So this was already 4 years ago. And yeah, we are kind of happy to see that this will also then be again used in Slovenia for such purpose.
Yeah, I know. I'm also glad. I was not really allowed to really mention that you really contributed to DTR, but now it was established in Avexis and now being fully validated and used in the GMP environment for release of the of this drug. But it's really a nice and great contribution of a Slovenian scientist. Yeah.
Yeah.
Did you guys develop empty full ratio methods that are validated, qualified?
Not yet. We are working on this ratio, so how to best determine them. But yeah, here the validations are a bit more tricky.
Yeah, I mean, that's what we are struggling with, and like everybody's doing things slightly differently. There's kind of 3, 4, 5 different methods that you can use, and they all show different thing on the same prep. So really kind of choose the right thing, and to really support your product is difficult, and the agency wants it, right? They really want to for you to have that part of the product. Because you can, like deliver potentially something to the patient that is like 50% empty, which is really something that should not be really released, right? In the first place. Even though it clicks all other specifications, right?
Yeah, well, definitely one really good approach is electron microscopy, because there you see the particles. With none of other methods that are used, you don't see particles as such. So you're just kind of indirectly measuring something. So this technique can give you quite a good insight in what kind of sample you have, but then validating that you're using the right or quantifying this ratio, this is a bit more problematic.
That's impossible to validate, but it's, I'm glad to hear that we're all struggling with the same problems, but I think like this is a good, it's really good to talk and to have kind of consensus of what's worth doing and what's not worth doing. I think at the end, science always wins, because you have to do the right scientific thing for your product, because that's the only thing that's going to work for the patients, right?
I think what we also already faced, and especially Robert, we have to educate also agencies, because the problem is that this is so specific knowledge. We cannot expect that they know everything. So we have to, and here is, I think, also very important position of the institutes, which have no conflict of interest, because they are, let's say, scientific ones. They are not companies. They are not living out of this directly, and they can, let's say, develop and support such agency with the knowledge and training for the specific fields. Otherwise, these fields are dead, because you cannot go through certain methods which are developed for the pharmaceutical products, which were normally one molecule, and even with the validation of this, you have to spend a lot of things, because you have a multilateral effect of these drugs around. And when you have a therapy or drug which is changing all the time, it is like a selection of the cells of the cell sorter. It's the same thing. You know, at the end, you get some cells which is completely different than it was in the beginning. So this is something which we are facing all the time. Okay, so we have 5 more minutes. I would like to give more comments also to Boštjan and Uroš. They're both technologies are those which are coming they are now more in research, but their therapeutical application will be seen soon. So first, Uroš, that's what we started in the beginning. Where do you see the, let's say, the entering of the of your technology, multiplex CRISPR-Cas to the real therapy? Where do you expect? It's like a prediction.
It has to do, I guess, with the development in genetics first, to be sure that if you target more than one gene, let's put it more broadly, and to know that this combination of genes will actually be specific for a disease, for a symptom that you're treating. And maybe I can relate this to what Vita was talking about, so to like personalized medicine, because I think that the first examples will be variants of monogenic diseases with other combined variants that will affect how this known mutation actually affects disease, how it is pathogenic, and then you can introduce then another, like maybe like have a combination of 2 changes at the same time. Probably it's all ex vivo, because it's for the foreseeable future. I'm not sure that this type of CRISPR technology will be available to treatment of like organisms, human level, the whole body level, so like ex vivo therapies. And yes, I would guess personalized medicine for something that we now call monogenic diseases, and it's actually monogenic disease with other variants affecting them. Okay. I hope that answers the question.
Yeah, absolutely. So, and I would link it to Boštjan, because that's then a logical thing. Then you make a tissue let's say, ex vivo transduced or transfected using, let's say, electroporation if we can not use a viral approach, and then the next step is that you are making a tissue. So what do you think about it, Boštjan?
Yeah, absolutely. So I mean, in the, let's say, the really long-term goal is to have the capability to make replacement tissues for regenerative medicine. But even before that, I think it's going to be really interesting to see in vitro tissue models to be tested with different drugs, because I believe that once we have the 3D biology to the level of repeatability and control, that there will be really good replacements for, let's say, animal models and standard 2D cell culture models. So this is definitely going to be interesting. Also, as I mentioned shortly in the presentation, our department works a lot with primary cell cultures, which we get from the clinic here in Maribor, and this is definitely also a good opportunity to go into personalized medicine in terms of getting, you know, tissue samples or biopsies from the patient, and then trying to build an in vitro tissue model from that you can then test with drugs, for example.
Okay, so, and then the last question is for Jelka. Do you expect that such tissues, or let's say somehow genetically modified cells, would be more immunogenic?
Yeah, that depends on how they are produced. I mean, it's, for example, for what we used in mice, for example, it's very important that you don't introduce some antigens that are not so, because you can introduce TCR and then we also modify them to express some other molecules. It's very important that they are not from another, from rat or from human, for example. So like this, you can then, they can be rejected, for example, such cells. So I think the, like, it was mentioned many times today. I think this CRISPR-Cas technology and electroporation are quite promising technologies to further develop this kind of cell gene therapies, because yeah, you will get less. Yeah, we should strive to get less immunogenic cells, and of course we have to test this before this comes to the clinic.
To the clinic, yeah.
We will have
Okay,
to wrap
yes,
up.
now we have to close. I will give just the closing remark before going to back to Gabriela. So thank you to all speakers and for the nice presentations and very important topics. And I'm glad to see how many different technologies from the beginning to the therapeutical approach are in our neighborhood. And that's also a call for collaboration, because just together we can do bigger steps forward, and talk also with big pharma, so which representative which we have here. So my message would be, our body does not consist of drugs but of cells. So please take this as let's say as an information for development of the new therapies. Thank you everybody, and Gabriela, now your turn.
Thank you. Thank you everybody. Thank you, Miomir. It was very useful. It was a very nice discussion. I hope this discussion can continue in Huva. I want to thank you, thank for your patience during the mail trafficking on the previous days. So we are finishing now. Please follow what's going on the Huva platform, where you can still meet with other speakers and get to know other people and discuss further the subjects that you opened here among yourselves. The link is in the chat box that will appear now on the Zoom chat box. We hope you enjoyed this event. Please give us your feedback. let us know how did this feel, how useful this is for you. Don't forget that this festival does not finish now. It is going on throughout November, so the next event will be already on the 17th of November on Smart Future. And you also can tune into the live stream of the Rector's Award ceremony for the best innovation of the University of Ljubljana. And also the companies are kindly invited to our bonus workshops that the researchers are offering as part of this UNI.MINDS festival. The next will be already tomorrow on cyber safety. And today at 12 o'clock, there is the Novartis Research Day. So if you have time, please, it's at 12, it's within half an hour. Thank you for everything. It was very nice. Take care. And let's all keep an open mind and continue with these collaborations that you already had and make new ones and feed your mobility and create a healthy future as it is the point of our society, I think. Thank you, everybody.
Thank you.
Bye.
Thank you. Bye.
Thank you. Bye.